Inverse PCR. It is also one of the variation of the basic reaction. And here different steps like the circularization of molecule, and then again digestion it is involved. Why we do all these steps? So actually this inverse PCR it is used to amplify or to know the sequence of the unknown region, and it is possible when within a particular fragment the sequence of one region it is known. इसको हम इस तरीके से सपोज कर सकते हैं कि हमारे पास जीनोमिक डीएनए है, हमने एक ख़ास रिस्ट्रिक्शन एंजाइम को यूज़ करते हुए उसको डाइजेस्ट किया, और डाइजेस्ट करने के बाद हमारे पास एक रिस्ट्रिक्शन फ्रेगमेंट आ गया. लाइक वी कैन कोट द एग्जांपल ऑफ दिस दैट इट इज़ अ रिस्ट्रिक्शन फ्रेगमेंट. नाउ विदिन this restriction fragment that has been digested with certain restriction enzyme, now it is with us. And suppose here within this center the sequence of the region is known, but this may not be our sequence of interest. Our sequence of interest may be the flanking regions that is present right or left to the known sequence. Now how we can know the sequence, or how we can amplify this unknown region, because we don't have primer that can bind with 3' or 5' end of the restriction fragment. How we can amplify this fragment? It can be possible by using a variation of PCR which is called as the inverse PCR. How? So suppose this fragment it has been digested with particular restriction enzyme, like restriction enzyme any, like 1 A, B, C, or it may be EcoR1, so that it has generated sticky end. Now with the help of those sticky end, this DNA fragment it can be circularized, convert into circle. So when it is converted into circle, now within the known region we know the sequence, we know the restriction enzyme which can be used that has the target site within the known sequence. We can digest it with the second restriction enzyme, so that cut is within the known sequence whose primer we can design. And after that, after circularization, again it is converted into linear form because of second digestion, and by using known sequence we can develop the primers, and those primers they can be further used to amplify our unknown region that may be of our interest. For example, in this case, it is example of inverse PCR, or the basic strategy that we will use in inverse PCR. It is a restriction fragment. This is the total length of the restriction fragment that is available. As I told you that one region may be having known sequence here. Here in this case, it is having known sequence, and this shaded region. And we can also develop the primer because the sequence is known, like the primer 1 or primer 2. Now how we will amplify this flanking region that may be of our interest? So we can introduce circularization of the restriction fragment. Why? Because sticky end is there because of the digestion with restriction enzyme 1 here. So circularization is there, and after circularization it is again cut with restriction enzyme 2 here, so that again it is converted into linear form. Now we have the sequence that are known at the ends, like here we can bind first primer, then second primer, and we can then continue the amplification of the region which is unknown. So by digesting the known sequence we can inverse the DNA target sites. So because of this reason it is called as inverse PCR, and this inverse PCR it is used to amplify the sequences of the unknown region.